Chemical carcinogens are substances that can cause cancer. They are a major public health concern. Many of these chemicals are found in everyday products. Some are even present in our environment. Understanding their dangers is crucial. It helps us make informed decisions. It also guides regulatory actions to protect public health. These carcinogens do not always show their effects immediately. Sometimes it takes years for their impact to become evident. This makes studying them even more important. Early detection and understanding can save lives. It can also reduce the burden on healthcare systems. This essay will explore 3 specific chemicals: quinoline, styrene, and styrene 7,8 oxide. The International Agency for Research on Cancer, or IARC, plays a key role. It evaluates the carcinogenicity of various substances. In March 2018, the IARC Working Group held a significant meeting. They reviewed several chemicals including quinoline, styrene, and styrene 7,8 oxide. Their findings are crucial for public health policies. The IARC uses rigorous scientific methods. They review studies from around the world. This ensures a comprehensive evaluation. Their classifications help in understanding the cancer risks. These findings are used by governments and health organizations globally. They form the basis for regulations and safety guidelines. The March 2018 evaluation brought new insights. It highlighted the potential dangers of these chemicals. This essay will discuss these findings in detail. We will explore the evidence and what it means for public health. Understanding these evaluations helps us grasp the seriousness of the risks involved. Quinoline is a chemical compound. It is a colorless, oily liquid with a strong odor. It is used in the production of dyes, antiseptics, and other chemicals. Quinoline is also found in coal tar. This makes it present in the environment, especially near industrial areas. Quinoline can enter the body through inhalation, ingestion, or skin contact. Once inside, it can affect various organs. It is metabolized in the liver and excreted in the urine. The concern with quinoline is its potential to cause cancer. Studies have shown that it can induce tumors in animals. The presence of quinoline in the environment is a concern. It can contaminate water and soil. People living near industrial areas may be at higher risk. This makes understanding its effects even more important. The next sections will delve into its impact on animals and the lack of data on humans. Animal studies are crucial in understanding carcinogens. Quinoline has been tested on various animals. These studies have shown alarming results. Quinoline can cause tumors in multiple organs. This includes the liver, lungs, and kidneys. These findings raise concerns about its safety. The mechanisms by which quinoline induces cancer are still being studied. It is believed to cause DNA damage. This can lead to mutations and uncontrolled cell growth. The extent of its carcinogenicity varies among different species. However, the evidence is strong enough to warrant caution. Long-term exposure to quinoline has shown a higher incidence of tumors. This is particularly evident in rats and mice. These studies help in understanding the potential risks to humans. However, translating these findings to human risk is complex. It requires further research and careful analysis. Section 5. Scarcity of human data on quinoline. Despite the strong evidence from animal studies, human data is scarce. There are very few studies on quinoline's effects on humans. This makes it challenging to assess the real risk. Most of the data comes from occupational exposure. Workers in industries using quinoline may be at higher risk. The lack of human data highlights a significant gap. It underscores the need for more research. Understanding how quinoline affects humans is crucial. This can help in developing safety guidelines. It can also inform regulatory actions to protect public health. Epidemiological studies are needed. These studies can provide insights into the real-world impact. They can help in understanding the long-term effects. Gathering this data is essential for a comprehensive risk assessment. It can also guide public health interventions. Section 6. What is styrene? Styrene is a widely used chemical. It is a colorless liquid with a sweet smell. It is used in the production of polystyrene plastics and resins. These materials are found in many everyday products. Examples include packaging, insulation, and disposable cups. Styrene can enter the body through inhalation, ingestion, or skin contact. It is metabolized in the liver and excreted in the urine. The concern with styrene is its link to cancer. Studies have shown that it can cause tumors in both animals and humans. This makes it a significant public health issue. Styrene is present in the environment. It can contaminate air, water, and soil. People living near industrial areas are at higher risk. This makes understanding its effects even more important. The next sections will explore its link to various types of cancer. Section 7. Styrene's link to lung cancer. Lung cancer is one of the most common cancers. Styrene exposure has been linked to an increased risk of lung cancer. This is based on both animal and human studies. Inhalation is the primary route of exposure. Workers in industries using styrene are at higher risk. Studies have shown that styrene can cause lung tumors in animals. These findings are concerning. They suggest a potential risk to humans. Epidemiological studies have also shown a link between styrene exposure and lung cancer. This underscores the need for caution and protective measures. The mechanisms by which styrene causes lung cancer are still being studied. It is believed to cause DNA damage and mutations. This can lead to uncontrolled cell growth. Understanding these mechanisms is crucial. It can help in developing preventive strategies and treatments. Section 8. Styrene's link to leukemia. Leukemia is another cancer linked to styrene exposure. This is supported by both animal and human studies. Workers in industries using styrene are at higher risk. Inhalation and skin contact are the primary routes of exposure. Studies have shown that styrene can cause blood cancers in animals. This includes leukemia and lymphoma. These findings are concerning. They suggest a potential risk to humans. Epidemiological studies have also shown a link between styrene exposure and leukemia. This underscores the need for caution and protective measures. The mechanisms by which styrene causes leukemia are still being studied. It is believed to disrupt normal blood cell production. This can lead to uncontrolled cell growth. Understanding these mechanisms is crucial. It can help in developing preventive strategies and treatments. Section 9. What is styrene 7,8 oxide? Styrene 7,8 oxide is a metabolite of styrene. It is formed when styrene is metabolized in the body. It is a colorless liquid with a strong odor. It is used in the production of resins and plastics. The concern with styrene 7,8 oxide is its strong evidence of carcinogenicity. Styrene 7,8 oxide can enter the body through inhalation, ingestion, or skin contact. It is metabolized in the liver and excreted in the urine. Studies have shown that it can cause DNA damage. This can lead to mutations and cancer. The evidence is particularly strong in animals. The presence of styrene 7,8 oxide in the environment is a concern. It can contaminate air, water, and soil. People living near industrial areas are at higher risk. This makes understanding its effects even more important. The next section will explore its carcinogenicity in animals. Section 10. Styrene 7,8. Oxide's carcinogenicity in animals. Animal studies have shown that styrene 7,8 oxide is a potent carcinogen. It can cause tumors in multiple organs. This includes the liver, lungs, and skin. These findings are concerning. They suggest a significant risk to humans. The mechanisms by which styrene 7,8 oxide induces cancer are still being studied. It is believed to cause DNA damage. This can lead to mutations and uncontrolled cell growth. The evidence from animal studies is strong. This has led to its classification as a probable human carcinogen. Long-term exposure to styrene 7,8 oxide has shown a higher incidence of tumors. This is particularly evident in rats and mice. These studies help in understanding the potential risks to humans. However, translating these findings to human risk is complex. It requires further research and careful analysis. Section 11. Conclusion. The need for further research. The carcinogenic potential of quinoline, styrene, and styrene 7,8 oxide is concerning. The evidence from animal studies is strong. However, there is a scarcity of human data. This highlights the need for further research. Understanding the risks is crucial for public health. Regulatory actions are needed to protect people. This includes workers in industries using these chemicals. It also includes the general population exposed to environmental contamination. Gathering more human data is essential. It can help in developing safety guidelines and regulatory measures. Public awareness is also important. People need to understand the risks. This can help in making informed decisions. It can also drive demand for safer alternatives. Continued research and vigilance are key to addressing these hidden dangers. Together, we can work towards a safer and healthier future.